Buenos días a todos y gracias por estar con nosotros en el seminario de hoy. Y voy a cambiar a inglés. It's a pleasure for us to have here Dr. Hugh Nicholas from Pittsburgh Supercomputer Center, a long-term friend of the Medical Sciences campus and of this department, and a collaborator also. He has been working closely with Dr. Ricardo Gonzalez, and has offered previously several seminars and workshops at this campus. Dr. Nicholas obtained his bachelor degree from the University of Oklahoma in chemistry, and then a PhD from the same university in physical chemistry. He has been at the University of Oklahoma as well as the University of Wisconsin, and in 1992 he moved to Pittsburgh Supercomputer Center as a senior scientific specialist. He's very well known in his field, has numerous presentations in well-recognized journals, supporting his research funded by NIH. Dr. Nicholas is going to talk to us. He's here also because he's a member of the thesis committee of one of my students, Rosana Cintron, who is presenting this afternoon. I almost forgot. It's a collaborative work with Dr. Ricardo Gonzalez, Hugh Nicholas, Roxana, and myself. And the title of the seminar is Changing Substrate Specificity by Remodeling Domain Interfaces in Glutathione Transferases.
Okay. Thank Adelpha for that flattering introduction, and say I'm glad to be back here again. And with that, I guess we will get started on the seminar. Okay, we're going to sort of address three questions today, looking at computational techniques that can provide light on the answers of three questions. The main focus is on this third question. The first one is just the basic biochemical question of how are enzymes physically organized to carry out a single specific function. And we'll see how some computational techniques that were used in this study can illuminate that question. And then the second and third questions are going to be the main focus of the talk. That's how do you alter an enzyme so that it carries out a related function that's however distinctly different. And in this case, we're talking about different classes of substrates. And we're going to try to answer this question using computational techniques. And so what we need to do is think about what are the defining features of defining things like substrate specificity in different enzyme subfamilies, and how can we create a model of that we can put into a computer program so that we can detect and identify these defining features of the differences. And we'll give a little bit of that. But most of the talk's actually going to focus on how we after we've detected these differences, what are they and how do they actually influence the structure and behavior of a family of enzymes. We're going to run through this, and in many ways, I think you should think, even though we're using a computer, it's very much an experimental investigation into real data. And to do this, just like in the lab, like you have to do in the laboratory, we have to define and create some kind of experimental system. It's not quite like a cell-free system for protein synthesis or something like that, but nonetheless, it requires a considerable amount of care and work to to create that experimental system. That experimental system in this case is what's called a multiple sequence alignment. And I say it requires a good deal of effort because we're not just going to run this through a single program because none of the programs that create multiple sequence alignments create one that's of high enough quality and reliable enough to do the kind of investigation that we want to do on that alignment. And that's one of the things, well, that Roxana spent a lot of time on in the last year or so is working with that alignment. And we'll probably hear more about that as she presents this afternoon. But anyway, what will go into that, creating this multiple sequence alignment, is the output from one or more alignment programs, programs for finding distinctive patterns in the sequences, and also our knowledge of the biochemistry and structure of the enzymes. All of these will need to be integrated into creating the multiple sequence alignment we're going to use as data for analysis of the subfamilies. And once we have the experimental system, we need to identify and make sure we understand its components. And by its components, I mean the groups of sequences that constitute an enzyme subfamily. The classical examples of these are maybe different hemoglobins, the alpha and beta hemoglobins that form the dimer in an active hemoglobin molecule, or a myoglobin that forms a monomer rather than the tetramer. And we use several sources of information for doing this. One is, of course, experiment. We have experimental observations from the laboratory about different substrates that some groups recognize and others don't. And then we're going to do a phylogenetic analysis and a principal components analysis to make sure that we understand which individual sequences belong within each of these experimentally defined groups. And then we can apply the model we have of these of defining features to each subfamily and identify what they are, and go back and integrate that with the with known structure of the subfamily and see how that actually affects the biochemistry. And this is the family of enzymes we're going to look at today. It's a very large enzyme superfamily. It's been a focus of quite a bit of research lately. A lot of environmental chemists are very interested in it. It turns out to be the family of enzymes that bacteria have adapted to metabolizing things like creosote in railroad ties and telephone poles. It's also important in drug resistance in humans. It causes human beings to excrete at a more rapid rate chemotherapeutic agents, for instance, and so people want to inhibit that. And it's involved in drug resistance in disease vectors, pesticide resistance in agricultural pest and herbicide tolerance in plants. And so fortunately we have lots of data. I have included about 250 sequences in the alignment I'm working with. If I restarted this today, there's probably 10 times that many available. I don't know that I'd want to put all that many in one alignment, but so there's plenty of sequence data available, and there's 50 plus structures available from different subfamilies. This is a rough three-dimensional ribbon diagram of a glutathione transferase. So you can see it's a dimeric enzyme. This is one dimer, this is the other one in red, and this is sort of a view that we will look at. Each of the monomers is composed of two domains. There's one domain over here, and the other domain, the domain interface is roughly along this line, and the other domain over there. The whole thing organized around a two-fold axis in the center there. This is just an example of what I mean by a multiple sequence alignment, and the types of things that you can find out about it. These positions highlighted in red, for instance, indicate things that are probably important for the entire family of particular enzymes that are that are under study. These two may represent, for instance, a disulfide bond that helps maintain three-dimensional structure. Proline that's present in all of the sequences may mark the end of some secondary structure element or a particular kink in a loop somewhere that needs to be preserved. What we're particularly going to be looking at is things sort of like I've highlighted in the grape color here. Things where the identities of the amino acids change along subfamily boundaries. Each of the subfamilies is marked by a different color over here. This is just a small example in the ones we actually use. We have multiple sequences in all of the subfamilies, but this is sort of to illustrate the idea that one subfamily will be represented by several sequences and another subfamily by several sequences, and we will look at things that might distinguish one subfamily from the other, or might distinguish one subfamily from all the rest, like this tyrosine-leucine pair right here for this particular subfamily is quite different from the amino acids present in the other subfamilies. And that kind of information allows us to at least entertain the hypothesis that something unique in this subfamily may correspond to that dipeptide presence in the sequences. And that's the kind of thing we'll want to model in order to look for those things. This slide is it's a simple example I use for undergraduate classes as an exercise in learning how to create good, high-quality multiple sequence alignments. It started off, it's in pretty good shape already. What we have here, sort of as a schematic, the double bars represent places where there are amino acids present in the sequence. Each single bar represents a gap that's been introduced by the global alignment program to compensate for the amino acids that are present in this particular sequence in this case. And the colored highlights are highlights from the pattern finding program that I like to use called MEME. And what the pattern finding program does is very much illustrated in this case by this pink bar. That is, it found a set of amino acids that were present in every sequence in this particular data set that the multiple sequence alignment program aligned exactly with each other so that the ends of the patterns lined up and there were no gaps introduced. Now, we have other cases like this red one over here where nearly every sequence has an example of the red pattern. However, one of them got moved out here completely out of alignment with these others. There's some gaps introduced in here. This region looks pretty good, but there's different gappings down here. And what's involved then is going through and looking at all of this in detail and trying to consider the evolutionary and biochemical implications of these gaps and where it's been put, and say, what should I move? Does this actually belong aligned over here with this, or should all of this be put together and shoved over here so that this red block is aligned with these and this brown block is over here with these? That's the kind of decisions one has to make where your scientific judgment very much comes into play, and your knowledge of what sort of amino acids are likely to align with each other. It's pretty rare that you get more than one of these that is as nicely put together as this pink one. More of them are like the red one or the yellow one in here, where you can see lots of individual decisions that you have to go through and look at the data and think about carefully before you're able to go ahead with either a good phylogenetic analysis or the analysis looking for distinctive group features. This is the set of patterns that we found in our glutathione transferases, and I've highlighted two sets. The set highlighted in red are those that are present in virtually all of the sequences, 242, 243, 204. This one is sort of almost systematically absent in one subfamily, 241 for this one. And these in cyan are present in only one of the 8 different subfamilies that are included in this analysis. And well, these in particular are very useful about confirming membership in a particular subfamily and helping you organize the alignment according to subfamilies, which we'll see. The start of the process here, these are the 4 what I call universal mean patterns that apply virtually all sequences. These over here are in the first domain of the protein, and they actually constitute the glutathione binding site. These are in the second domain and constitute sort of a structural core to that second domain that packs up against the first domain to create the interface between between the domains and between the subunits. The domain interface is particularly where the co-substrate that's going to be conjugated to the glutathione binds in this subfamily. And one of the interesting things that came out about this, when we first started, you can see this little yellow bar here. That represents, in most cases, a dipeptide that's either a phenylalanine and tyrosine, or a tyrosine followed by a proline. And that was sort of all over the place, but by using this pattern analysis and asking what's most conserved, we discovered that follows all the way through, and we'll see that actually is an important feature in defining sort of the structural core of this second domain. And in this view, where we've added all 20 of the motifs to the original, or added the other 16 to make 20 out of the original 4, it helped us decide how to align this brown motif, because that aromatic proline site was right here in this brown domain, and so we could see that it helped us make the decision that it belonged lined up like this, and helped us refine that part of the alignment quite a bit once we discovered that. And that FG actually can be discovered by looking at structural, or FP can be pretty much identified by looking at structures and thinking about them. And we'll come back to that point in a few slides. Also notice that once again, in this case we have this orange motif that that actually covers 4 of the different subfamilies. This green one that covers only one subfamily. There's some others over here that cover only one subfamily, and these, for instance, are unique. These 2 are unique to the alpha subfamily, and we'll come back to those in a little while. After we have the alignment refined, we sort of look to see what are the most highly conserved residues. You can get some of this information in initial stages, but it's likely to be incomplete. After you have the alignment refined, it'll be much more complete. And we find, once again, a couple of regions of highly conserved amino acids in the glutathione binding motifs, 2 motifs that make up glutathione binding site, and out here in the second domain, a little more dispersed set of conserved amino acids that once again are going to sort of come together in 3 dimensions to make up a conserved core for that domain. If we look at the different meme Zoops motifs, the 20 that we had, they fall into a consistent pattern that you can make what's called a decision tree with. That is, if the presence of these 3 really completely universal motifs indicates it's just one of the 8 classes that we looked at. But if you find motif 1 and 10, then you've reduced it. They cover, those motifs cover these 4 classes, so you know if it's one of these 2 that you have to be in that class. On the other hand, if it has motif 6 in, then it's not one of this class, it's one of those 4 classes. And similarly, you can keep following it. If it's motif 15 and 20 are present, then it belongs to the alpha subfamily. If 14 and 18 are present, it's either mu or pi, and if 5 and 7 are also present, then it's the mu subfamily. So we can use that motif information to make a robust classificatory system for the different subfamilies, at least for most of the subfamilies. Okay, so once we have the a good alignment, then we want to sort of refine our notion of what are the subfamilies, and the classical place to start that is a phylogenetic analysis, and this sort of is a rough picture of the phylogenetic analysis, where you can each of these names represents a terminus that actually represents several sequences, all the sequences that belong to that particular subfamily. And you'll notice I have 1, 2, 3, and 4 different things labeled theta that were labeled in the literature and in our original annotation as theta sequences. These divided out into 4 separate places on the tree, indicating that the theta group's not really a coherent sort of group. And if we go back into some of the more recent literature that's not yet reflected in the databases, some people have started naming at least 2 of these theta subgroups as omega and kappa sequences. So there's some experimental evidence that does distinguish at least those 2 groups, and these 2 still are sort of largely undistinguished. I've highlighted down here with the yellow circle the 3 groups we're going to concentrate on, and the reason for that is this end of the tree was much better defined than this end. If we perform some statistical analysis on this, it indicates that these things are not the sequences in this end of the tree are not very strongly related to one group or another. They're likely to move around with very little variation in the sequence. And while these are relatively robustly defined, and so we can believe that these categories or subfamilies are strongly supported by the sequence data, and we can have some confidence in the analysis that we get out of those. And so we'll concentrate on that. We'll also, just as an example, pick this delta group, which is relatively well supported for this end of the tree, and contrast that with these 3. The other technique that I like to use for looking at groups is a principal components analysis, and in this case it produces coordinates in a 6-dimensional space, of which I've shown 2, which are the 2 dimensions that have the best resolving power, and it really resolves 3 groups more or less. We have the deltas over here. Would be a little without a little more concern, I'd be concerned about those 2. The alphas are very nicely defined up here. This group of mu's is pretty nicely defined. That group's, once you look at other separate dimensions, it really is pretty well defined. And in these 2 dimensions, these things are not particularly well distinguished from each other. Particularly in dimension 3, these 4 subfamilies are not distinguished at all. They're a little bit distinguished along dimension 2. But in higher dimensions, all of these subfamilies then go ahead and distinguish themselves fairly nicely. But that's one way to sort of look up at things and to see if maybe you've made a misclassification. I mean, this is sort of things, okay, I should go back and look at these as well as check other dimensions to make sure that these deltas really do belong with the others here. But once we've defined all of the subfamilies, then we want to start putting things into structural terms so we can develop some concrete hypotheses. And this is once again, the 3-dimensional structure in a ribbon diagram with one subunit up here, the other subunit down here. And this is domain 1, and this is domain 1 over here. I apologize, these 2 shades of red here and here didn't show up as distinctly as they did on some of the other slides. This is domain 2, this part, and that's sort of the way the major domains fit into here. This sort of fills in when you put the highly conserved residues on here, which we'll do here. It fills in and makes a fairly solid three-dimensional structure. That's the glutathione binding site. The blue represents one amino acid that's highly conserved, that is outside of either of the motifs. Not necessarily every highly conserved amino acid is part of a motif. But all of the two motifs and this highly conserved individual amino acid all come together to form a very compact, well-conserved structure. That's the glutathione binding site here in the first domain. And then the second domain is once again up here. When I put the well-conserved amino acids into that, what we can see, going back here, we have the last turn of this helix and this fairly complex loop out here that's highly conserved, and we have this one kinked helix. That's the other part of that. That's the yellow. That was the yellow domain that we showed in the alignment schematics. And when we add the conserved amino acids into that, we can see that we have on top of this one helix, right at the end of it, we have one highly conserved amino acid that's not part of that domain, that packs in with this glycine and tryptophan that's part of this complex loop structure, and it sits on top of this other helix. This all interacts with this helix that's in the yellow, and that's the proline. And down under here, you can see it a little bit on this side, is the phenylalanine or tyrosine residue that packs in against this helix that has the blue residue at the top of it. And all of this makes a fairly compact and well-conserved structure that's the core of the second domain that provides along here sort of where the binding site for the co-substrate resides, along the domain interface here between the glutathione binding site on this side and this structural core, and there the other substrate is going to be bound. This is just a quick look, sort of from the other side, I believe, that shows this is the glutathione in gray. It's contacting at the top of this helix. These conserved residues here at the top of the sheet that packs against the side of the helix. It's in contact with those residues in terms of hydrogen bonds and other van der Waals contacts. This sort of packs in there to help hold the sheet and the helix in the common orientation. And this is the others, the co-substrate sort of in this striped pattern laying back along in here. Okay, now that we've got the common structure defined, or the common a good multiple sequence alignment and the subfamilies identified, we're going to sort of model what's distinctive about the individual subfamilies. We're going to use what's called a callback Liebler distance, and basically this is well, it's the only equation I'll show you. What you can think of it is we have two different distributions of amino acids. One we're going to label P, one we're going to label Q, that are taken from two different parts of the phylogenetic tree. And all of these subfamilies, for instance, if we're analyzing the pi subfamily, all of these subfamilies will go into the Q subfamily. All of the sequences in this, all of the sequences in these subfamilies will contribute to the distribution of Q at a particular position in our alignment. And then we'll take all of the sequences in the pi subfamily to get a particular distribution P for what's in the pi subfamily, And what the equation that we're looking at actually does is measure how different P and Q are from each other. And it's basically saying that if these distributions are very different across this branch, we have evidence that there's different selective pressures in this part of the tree and in this part of the tree, and so that we've evolved some new feature here in this part of the tree. That's the analysis that we're going to be doing. We're going to present the results in two-dimensional plots like this, where things that are important for the entire family will show up in this part of the plot, things that are basically uninterested down here in this quadrant of the plot, and out here in this quadrant of the plot we'll have some positions that are important to a particular subfamily. And this one's for the alpha subfamily, and these are the positions that are likely to be distinctive for the alpha. Well, they are distinctive for the alpha subfamily. They make it stand out from the others, and thus they're the most likely positions to contribute to novel biochemical features. This is probably the one slide where we need to maybe dim the light so you can see the colors a little bit. The point of this slide is that we have in here and in here and in here again more or less 3 clusters of positions, 3 or 4 clusters depending upon how you want to cluster things, of positions that have a lot of these discriminatory positions very close to each other along the sequence. They're not sort of spread uniformly around the whole sequence. And this is going to correspond to also unique particular locations in the 3-dimensional structure. And well, as we get on into looking how these fall in on the 3-dimensional structure, we'll see that these clusters are located in ways that at least can be rationalized in terms of providing substrate specificity, and which it encourages me to have more confidence in the analysis than it would if these were sort of just randomly spread all over the sequence and all over the structure. The first subfamily we'll look at, and the lights can come back up now, will be the alpha subfamily, and we want to just put this up to recall there were 2 motifs in particular that the alpha subfamily had that were unique to it, as well as these individual positions identified by our analysis. There is one of the motifs, and the other one is back over here on the back side of the second of domain 2. The one is almost at the end of it's actually almost at the end, very end of domain 2, and it comes back and plops this helix onto the domain interface on top of where the substrate binds. And what's interesting is there's nothing really unique about this helix. It doesn't stand out from sequences in the other subdomains, but the other subfamilies, but this motif does. And if we look at the residues identified by our analysis, this glycine-proline pair is in the middle of that motif, and what I think is going on with that is that it puts this kink into this loop that's not present in the other subfamilies and redirects the helix rather than sticking out over here, more or less wagging in the solution, into the domain interface, where it takes up part of the volume that's normally occupied by the other subfamily, by the substrate, and thus the alpha subfamily binds smaller substrates than many of the other subfamilies. The other things that show up to be interesting are one of the clusters of specific residues in the alpha family sit back here in domain 1 and modify exactly how different elements of the glutathione binding domain interact with the rest of that domain. sort of reorienting the glutathione binding site within the domain a little bit. Then we have residues down here, and let me explain that. Back up and explain the color scheme here. I've colored domain 1 in cyan, domain 2 in green. The highly conserved residues are in the magenta in all cases. Those residues in wheat are these that are the unique residues to the alpha subfamily. The ones that are in wheat and space filling. The ones that are in wheat but a bond representation are those that are in contact with one of the wheat space filling groups across either, in this case, the domain interface or across the subunit interface. So that this interaction between this aromatic molecule and these side chains that it interacts with across the interface here and these here are basically involved in customizing, in this case, the subunit interface for the alpha subfamily. And in this case, they're involved in customizing the domain interface, which contributes to orienting the two domains relative to each other in the dimer. And this then quickly is the mu subfamily. Once again, we have some residues out here, sort of behind the glutathione binding site that are reorienting it. And then we have a set of residues right in the center around the twofold axis that reorient both the domain and the subunit interfaces here because they're at that intersection where the two interfaces come together. These are smaller amino acids than are present in the other subclasses, bringing the two, both the domains and the subunits closer to each other, probably restricting the space where the co-substrate can bind. For the pi subfamily, we have most of the interaction is out here on the domain interface, a little bit in the middle sort of the glutathione binding site, and sort of an unusual feature, one of these highly conserved residues in the glutathione binding site is usually a tyrosine. In the pi subfamily, it's a histidine that contacts with this helix across the subunit interface, so that you've made some modifications across here and across here in the subunit interface, along with these modifications to the domain interface for those three subfamilies. And what I'm proposing as a hypothesis, and we'll be trying to investigate this by both experimental and some other computational means, is that these interactions really are what control the orientation of the two domains relative to each other, and hence the interaction with the glutathione with the co-substrate. If we look at the other end of the tree, the delta subfamily, we can see that out in that part of the tree, the subunit interface is quite different. It's full of aromatic residues, making a much larger sort of gap between the two subunits. Not sure exactly how that fits in, but that's it's quite a dramatic difference between the ones that we looked at earlier. And thanks to these people for having probably listened to me way too long go on about this and how it was evolving, and making contributions and insights to it. And with that, I'll be glad to take any questions that from anybody.
Any question for Dr. Nicholas? I have a question.
Okay.
You mentioned, and we know that GSTs are involved in some organisms in resistance.
Mm-hmm.
Has you looked at any difference between the GST from resistance and sensitive by the human? Any
We haven't looked at that level of detail yet. In humans, the ones I'm looking at, for instance, it's the alpha, mu, and pi subfamilies that are involved in eliminating chemotherapeutics. And so this is part of the adjustment in that. The next thing is to collect what I guess you would call a subfamily. For instance, humans have 5 different genes for the alphas. And so we'd want to find particularly which one of those was involved in the drug resistance and compare it to the other 4 and see what the differences are in that. We haven't looked at that yet. But that's, we have some collaborators in the medical school at Pittsburgh that are interested in that question, and so we will be doing that.
Any other question for Dr. Nicholas? Well, thank you very much for a very interesting seminar.
Okay. Thank you. I suppose I should tell you, send all of your graduate students to Ricardo's course where they can learn to do this.